We are starting now. With you or without you.
Okay,
I will take care of that.
yeah.
John Savarin, you're not sitting down.
John. So you got us both.
I will start calling you by name. Let's close the doors, Mr. DeRosa. Thank you. Next up on our agenda, we have our Director of Community Services, Gina Sweeney. Gina is the heart and soul of the community, of the organization. She does all the support groups, she does all the community fundraising, she does so much. I don't know, I can't keep up with her. She's a good friend, she's a great colleague. Let me introduce you to Gina Sweeney.
Thank you, Elizabeth. Thank you. Well, they only gave me 15 minutes, so I promise that I won't bore you. And I do have note cards because I'm accused of being a gabber. So if I apologize if I look down. What a great conference so far. The morning has been absolutely fantastic, right? Great, and we have a fantastic afternoon for you all, and lunch was really good. Everybody enjoyed lunch? Yes, lunch was good, So for those of you that don't know me, I am also living with CMT, so I understand some of your frustrations, some of your worries, and some of your concerns. I was diagnosed with CMT when I was 14 years old. I am considered a spontaneous mutation, which means there's nobody else in my family that has CMT. So it is me. But besides the worries and the concerns, I think that there's two other major frustrations that we all share. One, hardly anybody knows what CMT is, right? No, it's very, very frustrating. And two, there's no treatment or cure for CMT. Right? So that's another major frustration. But I think that we can change that. So tomorrow morning, when you get up in the morning and you grab your cup of joe and your Sunday paper and you're flipping through it and you see an article written about a disease or a disorder and you think, man, I was just at that CMT conference. I wish that article could be about CMT. Or you're channel surfing and you see a news anchor talking about an upcoming fundraiser or a fundraiser that has just passed. And you think, man, I wish that could be for the CMTA. Well, we in this room and everybody else, we are a massive team, guys. We are a team. If we do not do it as individuals, who will? Not your neighbor, not your employer. The CMTA is doing everything we can to get the word out and get you that treatment, but we can't do it alone. Together as a team, we can, right? So, STAR was born, which gives us all hope. Everyone should be extremely excited about the direction that STAR has taken. But again, the CMTA cannot do that alone. We need you as individuals. The more support that we get by donating and fundraising, the busier the CMTA can get in finding you all that treatment. Let me put it to you this way. There's 2.6 million people living with CMT, right? Can you imagine if everyone gives the CMTA $1 a year? Should I do the math? You want me to do the math? How about this? Can you imagine if that 2.6 million people gave the CMTA $5? That's a latte at Starbucks. How much would that be? $13 million. Where would we be as far as a treatment if we all work together as a team? So, besides donating and besides donating and supporting the CMTA, there are so many other ways that individuals can get involved with what we're doing. But what happens is so many people sit back and think, oh, I'm one person. What do I have to offer the CMTA? If you stop and you think. When you're looking through that paper and you're thinking about, I wish that article could be about CMT, you know what? I think my cousin's buddy works for the newspaper article. I bet if I call them, they would put an article in the paper. Or, you know what? I've belonged to this club, or my employer does fundraising for nonprofits every year. I bet if I'd ask them, they would do it for the CMTA. Sometimes it's just you have to stop and you have to think and you have to ask, right? So one person can make a difference. It takes one person to pick up the phone and say, Gina, I need 30 to 40 brochures. What is CMT brochures? Can you mail them to me? You got it. It takes one person to step up and be a support group facilitator for the CMTA. It takes one person to hold a fundraiser for the CMTA, big or small. The CMTA has something called Circle of Friends. You can educate your family, your friends about CMT. You can fundraise for the CMTA without even leaving your home. So, one person can make that difference. These faces right here, this is the future generation of CMT. And my little girl is right in the middle. This is the future. So, it is November. What do people normally do in January? 1, they sit back, I'm going to make a New Year's resolution. I ask you to make your New Year's resolution in 2014 to jump on board with what the CMTA is doing. Because if we work together as a team, we can make our dream their reality, right? So, with that, and this is where I'm going to get my note cards, because I will go over on this part because I have a whole lot to say. 3 years ago, the CMTA decided to recognize one of our community members for their outstanding dedication to us as an organization. We have many community members. Everybody is doing their part. We wanted to recognize one individual for the Rebecca Sand Volunteer Year Award. I am here today to present that award to that individual. Aha, I had a little surprise. Nobody knows that. Ha, ha, ha. But not only do I have one award this year, I have 2. And you will find that they both have different skill sets as far as volunteering to the CMTA. So let me tell you why the award is the Rebecca Sand Award. Rebecca Sand was really one of the first individuals to step up and say, hey, woohoo, CMTA, I want to fundraise for you, I want to step up, and I want to spread as much awareness as I can for CMT. Rebecca Sand received the very first Volunteer Year Award from the CMTA back in 1990. So that's just a little bit of background on Rebecca. So I'm going to move to my next slide, but you're still not going to know who the first one is. Because this, as I said, there's 2 different individuals that are going to receive this award, and we did a junior along with a regular Volunteer Year Award. So this particular junior has appeared in our CMTA newsletter, I think, every single issue for the past year. They have given countless presentations in school about CMT. They have given countless interviews for newspaper articles. Their family has stepped up and supported this individual, and they have fundraised as a family. They went above and beyond in the fundraising. It's important for you to know that this individual has only been diagnosed with CMT a year ago. He is a spontaneous mutation, just like myself, and he's only 15 years old. So, Pat, Elizabeth, would you guys like to come up? Mr. Victorio Riki! He's blown away right
Thank you very much.
I would like would you like to say maybe 1 or 2 words?
Sure.
Okay.
Okay.
Not like putting him on the spot or anything.
All right. Yeah, my name is Vittorio Ricci, and I am 15 years old. And I was diagnosed a year ago, unofficially diagnosed. I don't know my type. There's no family history. And so really, it was probably one of the best things that happened to me because I lived probably 3 years of my life with the full symptoms, not knowing what was wrong with me. And I went through middle school, which was, for me, kind of a difficult period because I was walking funny. And I didn't know why. I thought I was just an outcast. I thought I was a lot different than everyone. And I didn't have a lot of self-confidence, especially in my athletic ability. So when I finally got diagnosed, where most people got really upset, I was really happy because I wasn't just weird. I had a purpose to it. So, thank you.
All right, thank you. I think he may be the future CEO of the CMTA. You guys just want to stay up here. Woo! Jill, would you like to stand up? This is Victoria's mother. And as a family, I would like you all to applaud them for everything that they have done. So, our next victim, this individual also has only been involved with the CMTA for a year. He has given the CMTA a fresh, new look. From countless brochures, the Awareness Month microsite, he has given us, as I said, countless hours, but he has become an incredible part of our team. He has shared his skill set with the CMTA, all pro bono. And I am very, very honored to be giving him this award. He's given, as I said, the CMTA just an incredible, incredible look. Susan, would you like to come up here with me? Is Susan not in here? No, that's okay. I don't want to hold anything up. So I would like to introduce to you Bob DeRosa from Atrix. It's funny. He wanted to help me with this PowerPoint. Wow. Almost started crying there. Would you like to say?
No, I wouldn't.
No, please. Please.
This has come as quite a shock. I like to be behind the scenes, but my company, Atrix, became sponsors about 16 months ago, and it's, I guess the best thing I could say, I'm always being thanked for, but I want to thank all of you because it's been quite an inspiration to me to be a part of this association. It really has. Thank you.
Bob and Victorio, just kind of after the conference, if we can snap a couple photos, and I do have the boxes for your awards, so when I come down, I'll make sure that I do give that to you, okay? All right, so I think I'm just about done, so I hope it wasn't boring, I didn't bore you too much. I want to leave you with one last comment. Again, there's my little sweet Riley. But life gives you lemons, make orange juice instead, and leave the world wondering how you did it. We all have lemons, right? I saw this and I said, yes, that's what I want to do. Guys, I do not want to leave just a mark. I want to leave a huge dent in the CMT community. And together we all can. I do not want my great-grandchildren saying, I got my CMT from my grandma, great-grandma. I want them to say that she joined the fight against CMT. And they can all look back and say, they all rocked it. So I hope you all join us in doing that. Thank you very much.
Dr. Shai and Dr. Zuckner, could you please come up?
Boo.
Oh my God, I'm like, where is he? Where is he? He's right in back of me. You're mean.
I've been there the whole time.
You're mean.
So I've been asked to introduce Professor Stefan Zuckner, and I've known him for a number of years, and you just have to give me a minute because I want to try to think of something nice to say about him. And I'm just going to tell you very briefly a little bit about Stefan, that he actually grew up in Eastern Europe, in Dresden, and when the wall came down, he moved to do, right from high school, to go into learn medicine in Germany. He became interested in genetics, and he came and did a fellowship in the United States at Duke, and he took on a rapid path to become a superstar in our field. The first thing he did was he identified the genetic cause of CMT type 2A. So for people in the audience with CMT 2A, the reason that we know the causal gene was because of Stefan. And we didn't make it easy for him because shortly before he found the gene, somebody else thought they had found it and published it, an incorrect version, so he had to fight that. And of course, as always, he got it done correctly. Stefan has continued to do work in 2A. He developed a mouse model of it. But in addition, he became really interested in genetic sequencing and gene identification. And he moved from Duke to Miami, and he's now the acting chair of human genetics at the University of Miami. He has identified many other genes, but what he's also done is he's revolutionized, at least in the CMT world, our whole approach to how we do genetic diagnosis, because he's really developed the field of next-generation sequencing in CMT. He's developed applications which allow doctors like me to look at all the genetic candidates for CMT patients right on our computer in our office with an application that he developed. And in just the last year alone, I think he's identified 10 to 15 new CMT genes. So he really is a part of the reason that we're able to say that we now know 70 different genes that cause CMT, and he is working with our whole CMT consortium and really giving the genetic backbone to the whole structure. So other than that, he's a terrific person, and I can't think of anything else nice to say about him, so here he is.
So, thanks, Mike. What can I say? I hope it is all true what you said. I need to fix this first here. 1 second. So first of all, it's great to be here. I had some really interesting discussions already, and such a great spirit here. And over lunch, I said, 1 second. I said I really believe things are changing rapidly in CMT in terms of what we know about CMT, how we actually could do the next steps to understand the mechanics of the biology, and also how to create treatments. So overall, I'm very optimistic that we will see therapies coming. And only about 15 years ago, when I went to med school, I can tell you. And I did a neurology fellowship. Nobody thought this would ever be possible to treat something like CMT, and I think this really has changed among doctors that there might actually be a solution in the future. So the bad news is when I came here on this short flight from Miami, I discovered that I lost my file. So in the last hour or so, I put together a new talk, and but that's also actually not so important what each slide shows. Some things might be a little bit technical, so just glance over it. I just see it as a kind of an overview what a guy like I do and how this might relate to CMT, and just enjoy it. So CMT is a genetic disease as far as we know today, and so I do study DNA, and this is kind of an example of what DNA looks like. What we think it looks like is the double helix, the famous double helix, and it's actually it's a very thin molecule. It's a continuous molecule. When you stretch it out, it's about 6 yards long, and you have a copy of that in each cell of your body. You have about 100 trillion individual cells in your body. Each cell contains the whole set of genetic information. And it was always a dream for biologists to be able to understand the whole DNA. And then in the late 90s, the US government decided to decode, read out the human DNA for the first time. It took 15 years. It cost $3 billion. And in 2001 was the first time this was finished. And this is the moment when Bill Clinton actually announced that the project is almost done or done. And quite interesting, even back then, that's now 10 years, 12 years ago, this is Francis Collins, who is now the director of the National Institutes of Health. He was leading the Genome project. And this is Greg Venter, who's a scientist who started his own private institute. And just with private effort, he achieved in a much shorter time what the government did in like 15 years. Now, I wouldn't say the government was inefficient, but it just shows you with the spirit sometimes that private enterprise brings to things. You know, this is also a power we should not forget. It's not just happening at universities, it's happening everywhere. I thought it was quite interesting. And then in 2003, the human genome was finished the second time, meaning we had a more refined version. And that is kind of the official end of it. So that really gave scientists a template to work off. And now 10 years later, you really see the fruits of it, because genetics has changed dramatically. This whole project, the 15 years of finding out the genome, was done with a technique called Sanger sequencing, which by today's standards is like dinosaur technique. But it's really only been 10 years ago that we used it, and we still use it actually. And because there was so much interest in sequencing of DNA, again, private industry came in, smart people got really interested in this field, and they developed a new way to sequence, to read out DNA, which we call next generation sequencing. A very innovative name, I guess. But so this new technology, the next generation sequencing, has really changed everything, and that's what we use today. And as a consequence, sequencing has improved by 100,000 fold in just 10 years or so. And that's way more than what we know from the Moore's law. Some of you might know the Moore's law in computing, that computing power doubles every 18 months or so. 100,000 fold in 10 years, that's way higher than that. 10 years ago, a genome of a single person would cost about $3 million to produce and take probably a year. Today, only 10 years later, it costs $5,000, and you can do it in 2 weeks or so when you're really fast. And there are other techniques that make it even much cheaper to look at people's whole genomes. So now we really can look at the whole DNA of a single person. So each one of us has 3.2 billion pieces of DNA. The DNA is made up of base pairs. We call them base pairs, and we have 3.2 billion of these. And they mostly, between any of us, they're mostly identical, as they should be, because we're all humans. But there are some differences, and some of these differences account for that somebody gets CMT or does not. And there are other differences, of course, as well. So we're about 99% the same. But 1% of 3 billion is quite a bit. So what genetics really does, we look for the differences. We try to understand what is this 1% difference? What is it? What does it mean? And so on. And so what does it mean for CMT genetics? First of all, of course, the technology has been developed for all kinds of reasons, for scientific satisfaction, to understand cancer, to understand autism. But coincidentally, it also helps us, the CMT community, because now we have this technology available. So what happens now, what we see is that medical genetics testing, diagnosis in CMT is really changing. I had a lot of questions over lunch about testing issues, and it's expensive still. It's not complete. You go to certain labs that we all know their name is, and you get maybe only 10, 15 tests for a lot of money. And this is changing as we speak. This is really changing now. There are already smaller companies, smaller laboratories. They offer maybe 30, 40 genes in one package for a much smaller price. And as I said, we can now do a whole genome for $5,000, a whole genome. Why should 15 or 20 genes cost $15,000? Makes no sense. It just takes a little bit of time to adjust, but I think in the next 2 years we will see much better testing come out, and for a much more reasonable price point. The other thing is that, as you know, there are many genes for CMT. And Mike just mentioned that we have 70 genes now. For CMT2, that explains still only about 40% or so of all patients. So the majority of CMT2 patients, we still don't know what their gene is. There will be several hundred genes in the end. And I think we will be able to test them all. So gene identification is a very acute issue we work on right now. And then we also look forward already, trying to understand why in one family, why is it that one person has a very early onset and another person, a sibling or a niece, has a much milder type of CMT. Even though it's the same family, they should share the same mutation and so on. So we call these modifier genes. Okay, so this is like the traditional approach. Again, the so-called Sanger sequencing, and there was the time when each of these bases here, G, C, C, A, would make a nice little signal. It was easy to read out. The downside is that, it takes a longer time. When you read out 3 billion of these, that's basically impossible. So on a larger scale, this is just not as efficient. And by the way, here you would see, normally you see just one color at a time. And over here is a mutation where you see two colors kind of in each other, a green and a black. I don't know if you can see that. But that's like the old way to do this. And we still do this sometimes today. Now, the new approach is the extreme is, of course, to say, okay, now we can do it. Let's sequence the entire genome. And that was done for CMT the first time about 3 years ago. And in fact, that paper, which came out in a very prominent journal, the New England Journal of Medicine, is one of the most famous journals in all of medicine. That was the first ever sequencing of a patient with a diagnosis, whole genome sequencing, in all of medicine. So you see, CMT is actually a pioneer. We're not just piggybacking on other things that seem more prominent. This was the first diagnosis done by a whole genome sequencing, and coincidentally, it was done in a CMT patient. And this is an example what whole genome sequencing can give you is they sequenced this person here and they identified the gene, and then they went back and sequenced only this gene in the other siblings and the parents, and they found, of course, as expected, that's a recessive gene, first of all, so you have to have two changes here two mutations, a red one and a blue one. And all these people here, they had 2 copies, so that's why they were sick. Turns out the father had only one change, and the mother had the other. So these changes came together in the children, in some of the children. But then they also saw, well, the father actually didn't have the same kind of CMT, but he had some other issues with the peripheral nerves. And that's kind of a type of medicine we will see more and more, that by knowing through whole genome approaches more details of a person's DNA, we can even explain minor changes or similar changes in the family. So in this case, even though this is a recessive gene, you need 2 copies to get the full picture. Turns out the father had some other problems, and he had only 1 copy. So in other words, we're still learning what all these genetic changes, what they mean. And I'm gonna skip a little bit over this. So I mentioned whole genome sequencing. There's another technique we call exome sequencing. And you might hear this when you go to your doctor or so, or you're involved in a research study that we can now do exome sequencing. What this means is basically, let me go back. With whole genome sequencing, you cover the whole genome strand with these red pieces where you measure what is the sequence at this position. With an exome approach, you actually focus more on the genes. You don't want to know all the things in between. We know in CMT the genes matter most, so you go straight for the genes. What it does for us, it's much cheaper. So we can do an exome screen for about $1,000 now. And I might not even go too much into technology here, but it's quite interesting. You know, it's almost like magic when you think what happens on these instruments who can measure this. It all happens on a glass slide. It's about, I guess, 2 inch by 1 inch. It's a regular glass slide, and the DNA is being prepared in a certain way, chemically prepared, and it's then put on this glass slide, and this glass slide is put in a machine, and the machine reads this glass slide, and there's a chemical reaction that happens over 2 weeks, takes about 2 weeks, and this produces, and it's a constant measurement what happens on the surface of this glass slide. And at the end of this, you have about 40 terabytes of data, and this is being processed in big computers, and then you end up with a single mutation. So it's quite amazing, actually, how it works. So the exome sequencing, again, is a more economic way to do things, and we do this routinely now in our research studies. And what we do really is we look at so we all have about 20,000 protein coding genes, and they make up about 200,000 smaller pieces, because genes don't come in one package, they usually come in smaller pieces. So we measured 200,000 positions in the genome in one go. And what you get out of it looks a little bit like that. So you remember when I showed you the Sanger sequencing, the nice waveform? It was kind of easy to read. Try to read this. You get a headache, right? But that's the new way to do it. And immediately you would say, well, but that looks like a computer could read this very well. And that's true. So really what we see in genetics, it becomes very computerized. Everything becomes like data. It's all about data now. It's not so much about printing a piece of paper and looking what I see. It's really zeros and ones in the end. And that actually is a great opportunity, I think, because there's a whole industry out there who learned how to create computers, make them better and better, and we can basically tap into this knowledge as geneticists. So I want to show you an example. That is a study which was one of the first studies who used exome sequencing for CMT. It might have been even the first family where we used this exome approach to diagnose a CMT family. And this is the family, which might look like some of your families. And we decided to exome sequence these 2 individuals. They were already excluded for some major genes for CMT. And we wanted to see if we can identify the underlying gene. And in fact, we did. We found that this was a CMT that was on the X chromosome caused by connexin 32, or another name for that is GHB1. And we confirmed it with this older technology, the Sanger sequencing. So we really were able to identify the gene there quite fast. And what I need to point out, though, is we produced a huge amount of data and did this big analysis, and then we come back to this single change. And that's kind of the difference now. You have to collect enormous amount of data, analyze them to really filter down to just this one change. But this is mainly automated, so it works very well. And when you do this and look at 200,000 positions in the genome, you find other things as well. So in this family, we found additional changes, many changes. This is just a few of them. And these are all genes that have been implicated with CMT or similar diseases. So we found all these changes, other changes in these maybe CMT genes. And that's kind of a new problem we have now. When you test more, you will find more things. That's true in life, right? So now we have to deal with this. We have to understand, what does it mean? We find all this additional variation. And that's one of the big challenges we have today with this type of new data. So when we do a lot of this genome-style sequencing, we have to deal with a lot of large data sets. It's harder for say, the non-computer trained person to actually read the data because it's also computerized. And I think it's very important to bring the international research community together in some fashion so that they can work together and they can see each other's results, and then they can make conclusions, and this will really speed up research. And this is how we came to develop some special software a couple of years ago. Now, I'm trained as a doctor, and I can kind of relate to this issue that you're just not a computer-trained person. And you will never just learn that piece. So I think the computer world has come to the non-expert and make it simple. It should be like Google, right? It should be simple. So, and that's kind of how we set out to do that. And we created a system where now people can send us data, we put it in the system, and then it's fairly simple to actually search it, as Dr. Scheid can tell you that it's true. So this is, just to give a brief idea, this is how this looks like. It has a very simple interface. Really, you just select what you want to do in a very simple fashion, and it's very fast and gives you results back. And it gives you additional information here that is maybe not so interesting for you, but for a physician or a scientist, this could give you additional clues if a variant you find in a patient has any relevance or not. So I'm going to skip over this here. This has been also, the system looks very nice, it's very simple to use, but it's also very fast. So basically, it takes often a month to enroll a patient in a research study, right? And to get the blood sample and so on. And it takes still weeks, at least, to produce this huge amount of data. But once it's in the system, the analysis is done in a second. And this is really fascinating. So what used to be just a couple of years ago a huge problem, now I have all this data, how do I ever analyze it? This is gone, basically. So we provide this software, and this is web-based, so all the researchers around the world can access it anytime. even from their smartphones. And they can look at a family in a second, and you see what is in this family, what is wrong. So this is really great. And because it's quite user-friendly, a lot of researchers have signed on now. Nearly 200 investigators around the world, not just working on CMT, also on other diseases, truly international. And they are sending us more and more data now. And the more data we have in the system, CMT data especially, the more connections we can make. And we might find that something from a CMT patient in Tucson, Arizona, matches with something a colleague in Japan finds. And because of this connection, we can actually make the right conclusion. And this is a breakdown of all the countries involved. And here's a real life example, a real story, to make this a little bit more clear. So about a year ago, somebody from Australia, Catherine North, Dr. North from Australia, she had a CMT type family, and they thought they had identified a novel gene there, but they had just one family, and with one family you never know if that's something that's true for the general population of CMT patients. So she looked in the database that we have, and within seconds they saw there are potentially at least 2 or 3 more families with this gene in the database. And at the same time, basically, Dr. Mary Riley from London, she had another family, and she came to the same conclusions, and she also contacted me. I was working with Mary on this anyway. And so basically within days, we could coordinate a collaboration, and we found additional families then within days. We had families from the US, from Canada, from Brazil, from Germany, from Austria, from Australia, and from the UK. So without even $1 spent, basically. And that's the new way to find these disease genes. And when we published this in one of the best journals for genetic research, the American Journal of Human Genetics, we saw that 2 other groups had also found at the same time, basically, this gene. So this gene BICD2 is now a major gene that causes spinal muscular atrophy, and also in some patients a form of CMT2. And so it's definitely a major gene, a new gene for this spectrum of patients. And these patients, they have also some characteristics. Their arms are fairly preserved. They have strength there, but you see that the legs, they have this typical CMT legs, but they also, in addition, they have they lose muscle in their upper legs, so they're also very weak in their upper legs. That is different from the classical CMT2. And examples like that happen now all the time. And as Mike mentioned, it's really true. We have about 10, 15 new genes that we found in the last 18 months, and a good number of them is not even published officially yet. So we're quickly expanding the number of known CMT genes, so that we go from this 40% of patients that we know can diagnose today, hopefully to something like 90% at least in the near future. And today we have 70 known CMT genes, as mentioned. We think it might probably end up with a few hundred genes in the end. And now, why is that important to know all these genes? Many of these genes are really rare. There are just a few families out there that we know about. But each gene gives us a kind of a keyhole view into what is wrong in general in CMT. And it's true that most of what we know about CMT today came through genetic discoveries. And even a gene that might affect only a few dozen people in the whole world, as far as we know, can give us very important information that is important for a much broader set of CMT patients. So that's why we are hunting down also these rare genes. So another thing I want to bring up is a phenomenon that I'm sure you have talked in your families about, is that there is a spectrum, of course, of CMT. As you know, there's the demyelinating form, CMT1. There's the other end of the spectrum, that's the CMT2 patients. And there's, in fact, also a group in the middle that we call intermediate CMTs. And it's been noted for a long time, actually, that even within the same family, with the same mutation, people can have different expressions of their CMT. They can be more severe, they can start earlier or later. And there are even families where one might have a classic, or at least there are genes that are associated with more classic CMT1 in most cases, but sometimes they also can cause CMT2. So we call this, there's a spectrum. There's a spectrum of what we say phenotypes, or a spectrum of disease expression. And we are starting now to design studies so we can better understand what that is. Where does it come from? It looks like a lot of this also comes from other genes, secondary genes that kind of modify how the disease will progress. And if everything works out, we envision a future where in 10 years you get a whole genome screen. It tells you what primary CMT gene you have in your family, but it also tells the doctor what secondary genes are there. And he might be able to make predictions for you personally how the disease will progress. And that might be a different projection for you than it is for your brother, who also has the primary CMT gene. So we want to refine our abilities to predict what's going to happen. And it might even lead to predictions about what therapy, once we have them, would be best for one patient versus the other patient. So we call this personalized medicine, and I'm sure you have heard about this. So that is what we try to do right now. And we have now in the middle of the first such studies in CMT to find such modifiers. It's been a huge international initiative that involves some 17 different universities from multiple different countries, from the United States, from countries in Europe, including the Netherlands, the UK, Germany. Also Korean samples are included in that studies. So we have now analyzed the first 500 patients, 350 are pending right now, and each one has been tested for 650,000 positions in their genome. We call this a genome-wide association study, or GWAS study. And this is the first GWAS study in CMT. It's been done in other diseases, but that's the first one for CMT. And what we look for here is they all have CMT1A, all 500 have CMT1A, which is caused by a common mutation that is a duplication of the gene. So they all have the same mutation. All 500 have the same mutation, causes CMT1A. Yet, some have severe forms of CMT, some have mild forms. And with this study design, we try to find these modifier genes. And here are the first kind of results. This is what we call a Manhattan plot, because it looks a bit like the skyline of a big city. And all these nice colors here are the different chromosomes. So there's chromosome 1 here, chromosome 2, 3, and so on. And what you see here, all these little dots, there are actually 650,000 little dots in this picture. And most dots are around down here, as you can see. But some of these dots, they are up here. And these are the signals that we're trying to identify. Because when the signal is high, like here or here, those are potential genes that actually are modifying genes for CMT. So this is still ongoing, this study, but very exciting. We found the first candidate gene, which is called neuregulin-1. And coincidentally, neuregulin has already been shown in animal studies that it interacts with CMT1A. So this protein is a partner, so to say, partner in crime with the CMT1A gene. So there, right there you see how we can actually potentially find these modifier genes. Okay, I have one more interesting story to tell you. which covers another aspect of what we do in Miami. That is, so we have studied this small family here, actually from the United Kingdom, and these two sisters here, they had a fairly early onset. They lost their eyesight, it's what we call optic atrophy, when they were still children, and later on they developed CMT too. And there is a subgroup of CMT patients, especially CMT2 patients, who can have problems with their vision. They can develop optic atrophy. So we are interested in this combination for sure. So we did a genome screen on this small family, and just with these two sisters there and their parents, we are now able to actually find a gene. This is how powerful it gets. In the old days, we needed big families and so on. Now we need just a small family like that to identify the gene. It doesn't work all the time, but we have a pretty good chance now. So the gene we found, a new CMT gene, has this wonderful name, and and this is a recessive gene. Again, you need 2 mutations. These are the 2 mutations we found here and here. And what we can do now is fairly rapidly, we can take this information and try to model the disease in a system that is the zebrafish system. So zebrafish are small fish, about 1 inch long. You can have them in your laboratory in special tanks, and you can actually breed them. And because they're small fish, they have a fairly rapid cycle from generation to generation. You can genetically modify them very specifically, and so it's a great animal model for us to study, and we use it for CMT research. And I try to convince you why this is actually an interesting model. So we took these fish, and what you can do really within weeks, you can do an experiment like that. You can take out that gene of interest. And I have a couple of videos here. That's always fun. So when you have a normal fish like this and you touch it, it would swim away as expected. This is the fish with this, where the gene is lost. And you see it can still move, but it's kind of weak. It cannot really move fast. And so this might be an expression that might look like CMT in a fish. Fish don't walk, so we don't know for sure. But what we know about CMT in people is that certain nerve cells are affected. As you know, the nerves in your arms and legs, mostly legs, are not, they have a problem. So in these fish, when you look at the nerves here, this is the spinal cord, and here are the nerves that come out of the spinal cord. This is pretty much are the same nerves that run into your legs. You see that this is a normal fish. These nerves have a certain length, and that might not look impressive to you, but that's how you know, when you measure actually the nerve length in these fish, it's significantly lower, it's shorter. So somehow they cannot grow their nerves that well than normal fish. And you can knock down this gene so the nerve is shorter, and then you give the gene back to the fish. And when you do that, it grows to a normal length again. So it's what we call a rescue experiment. So what we also did, we took a very close look at these nerves when they reach the muscle, the so-called neuromuscular junction. And when you do this is a normal nerve. It's very fascinating, actually. This is a little movie of a mature nerve touching the muscle. And you would think that this nerve, once it has established this connection, it's kind of a stable situation. In fact, these nerves, they're always active. And you see how this nerve always kind of adjusts by growing little extensions to the muscle, depending on the situation on this muscle. And in the sick nerve, you see there are much less extensions to start with, But what is interesting, it still tries to reach out to that muscle and make the right connections. So in a way, that's a good thing. There is an inherent ability of our nerves they want to try the right thing, even in CMT patients. But there's something that hinders them. If we can take out that what hinders this nerve to grow and keep these connections going, then we might actually kind of treat these fish. And these are the kind of the concepts we try to bring to people too. And so this is just a little peek into the future here. And I guess I'm going to finish right on time. And I want to thank all the many people involved. Probably too many to mention here, but it's a fantastic community, the CMT research community, and as I said, the future is bright. So if you have any questions, please ask me.
Thank you, Dr. Zuckner. We have a break now, so if you want to ask questions to Dr. Zuckner, he'll be here for a few minutes, I think. Would that be okay? People come up and talk to you?
Sure.
All right. And we have another break, so stretch your legs, because then we have a long talk by Dr. Shai coming up. So stretch your legs, get something to eat, and see you in 15 minutes.
Hey, give me 1 second.